everybody for joining us. Today we have Dr. Brittney Dickey. She is an assistant professor in the Department of Epidemiology at UNMC. She completed her MPH in health promotion and PhD in epidemiology at UNMC before moving to Moffitt Cancer Center in Tampa, Florida for postdoctoral fellowship in the Department of Cancer Epidemiology. While there, her studies focused on HPV natural history and cancer among people with HIV. It was at Moffitt that she also began work in molecular epidemiology and epigenetics. Today, her research interests include infectious disease and cancer, using tools in basic and molecular epidemiology, which include topics such as epidemiology of oncogenic viruses, biomarkers for early detection of cancers associated with a viral infection, genome-wide DNA methylation as a tool to understand cancer risk, HIV, and aging. Please welcome Dr. Dickey.
All right, thank you so much. I totally forgot that I gave you that intro information and I put that on my first 3 slides. So I will go over them quickly. So I wasn't sure who the audience would be today, and so I thought it might be pertinent to include a little background information about myself. While I am a new assistant professor, I have actually been with UNMC for a long time in various roles, which I kind of put here on my timeline. I was an intern. I worked in the Epley Center focusing on immunology. And then I received my MPH here in the College of Public Health, as well as my PhD in epidemiology, where I was a graduate assistant and worked with many of the professors here. And then, as Heather said, I went to Moffitt for postdoctoral training. I like to include this at the beginning when I explain what it is I do or like to do, because I think it's useful. So the cancer care continuum, if you work in cancer research, you're familiar with this. If not, essentially it just highlights key points along the care continuum, from preventing cancer to early detection of cancer, diagnosis, and then all the way through palliative care and end-of-life care. So my research really focuses on this front end of the continuum. I'm really interested in thinking about risk factors for cancers and how we can use that information for prevention or early detection. And again, because I wasn't sure who would be here, I included all of my areas of interest. Today, I'm going to just focus on one, but I have some work in cervical cancer screening optimization in global setting. I'm interested in that in Nebraska as well. Thinking about cancer and cancer screening among people living with HIV or other immunocompromised individuals. And then Heather said this already, but most of my work is focused on either oncogenic infections and their progression to cancer, which can include immune moderators of that progression, or cancer in the setting of immunocompromised individuals. Hopefully you're sensing a theme. So today specifically, I'm going to talk about oral HPV and oropharyngeal cancer. I've given this talk before, so I apologize if you've heard it before, but I think that this talk really sums up a lot of the individual pieces that I do. So thinking about epidemiology and using biomarkers and thinking about molecular epi, and hopefully you'll see that here. So just starting with a little bit of background on oral HPV infection. I don't know if you're familiar. So most notably, oral HPV infection is more common in men than women. And you can see the prevalence is 10% in men compared to 3% in women. So much higher. High-risk HPV and low-risk HPV are 2 designations that we give HPV. And essentially the high-risk HPV is 14 types that are associated with cancer. They're considered oncogenic. The low-risk HPV are the ones associated with warts and other lesions, not cancer. And so some of the early work that I did, I share it here over on the side, where we started looking at factors associated with high-risk versus low-risk HPV. And they do have some differences. Most notably, the sexual factors are more associated with high-risk HPV rather than low-risk HPV, but there are some factors here for low-risk HPV. And then these, I did not produce these lovely graphs on the side, but I use them because I think they denote how HPV looks different in men versus women, which is really an important thing to understand. You can see this nice bimodal peak for prevalence among men, and you see it a little bit here with women, but it's not nearly as pronounced. And sorry, this is looking at age on the bottom. And so what that tells us is we really have a high-risk group of older men who are at risk for oral HPV. And so why do we care about oral HPV? We care because it is a known cause of oropharyngeal cancer. And so I use this lovely graphic here to remind everybody exactly what oropharyngeal cancer is. There's a lot of small parts in, the mouth and the tongue. And so the oropharynx is really just the back of the throat. And in fact, it's the back of the tongue. The front of the tongue is an entirely different subset of cancers. And so oropharyngeal cancer has historically been associated with tobacco and alcohol use, but that has shifted in recent times, and now more oropharyngeal cancer is associated with HPV rather than tobacco and alcohol use. And in fact, they are considered two distinct etiologies. And something I like to mention is OPC diagnosis is really unique in that they will automatically, part of the diagnosis is to test you for HPV. If you are positive for HPV, you are staged differently than people with an HPV negative OPC, because we know the etiology is different, the prognosis is different, the survival is different. They really are distinct. And so in my work, we're obviously focusing on the OPC that is associated with HPV. And again, I would be remiss if I didn't include a little epidemiology here. There's some important information here, but the key takeaway is you'll see that just like oral HPV infection, OPC is much higher, occurs much more often in men, but it also occurs more often. This blue line here in the middle, you'll see it's predominantly occurring in white men. So the high-risk group for HPV-associated OPC is mid to late adult white males. And interestingly, OPC is projected to continue these trends that you see here and have over 30,000 new cases by 2029. So I like to show this graph when we think about HPV-related cancers because I think it surprises some individuals. And so you have some of the types that you might be familiar with that are associated with HPV. Vaginal has the lowest numbers, and then so on and so forth. And so when you think about what's going to fall in these bottom two regions, you might be surprised to know that cervical cancer is the next most common after anal, but that oropharyngeal cancer is now surpassing cervical cancer as the most common HPV-related cancer in the United States. And as I've alluded to before, the difference is really quite staggering between males and females. Ooh, sorry about that. And so risk factors for OPC, I kind of mentioned this already, but I wanted a clear-cut slide on this. There is HPV negative OPC, which is historically and still associated with smoking and alcohol, but more so smoking. And you can see a 13 times higher risk of HPV negative OPC among current smokers compared to never smokers. And then HPV positive OPC, not surprisingly, is associated with increased sexual behaviors. So risk is 4 times higher among those with more lifetime oral sex partners. Some studies have found things such as younger age of sexual debut and increased prevalence of oral sex to be associated with HPV positive OPC. And so I mentioned at the beginning that I'm really interested in prevention and early detection, that front end of the cancer care continuum. So I wanted to share a little bit about what prevention looks like for HPV-associated OPC specifically. I think the obvious one we all know about is the HPV vaccine exists. We know that it is used to prevent high-risk HPV infections and low-risk HPV infections. There are 3 types of an HPV infection, and the one you're probably most familiar with, which is what's currently used in the US, is the 9-valent. It prevents these 9 types of HPV. However, the quadrivalent and bivalent are still used quite a bit in lower economic regions and global regions where access is just a little bit different. So you might think, well, we have a vaccine, then what's the issue? We're just going to vaccinate. But right now, HPV vaccines are recommended for the prevention of cervical cancer worldwide. That the FDA is expanding their licensure to now include prevention of OPC. But as you can imagine, these vaccines were largely targeted to women and young girls. Now young boys are included in this so much more, but it's still just not where it should be. So we're on the right track to prevention with vaccination, but it's not the solution at this point. And so the next option is screening and early detection. And I bring these two up side by side because screening for an HPV-related oropharyngeal cancer is very different than other HPV-associated cancers. We know that you can screen women for cervical cancer, and you can diagnose what's called a precursor lesion. Before it ever becomes cancer, you remove that lesion and you're good. Same thing with anal cancer. However, there is no known precursor lesion for oropharyngeal cancer. So really primary screening is non-existent. The best thing we can do is detect it early. The earlier we detect it, you're obviously going to improve survival, but as you can imagine, the treatment for cancers in the region of the head and neck, things like radiation and chemotherapy, can leave some pretty devastating comorbidities. And so that's really a big goal of this. And so when you think about early detection, there's a lot to think about. Who do we target this early detection to? We're not going to go out and screen everybody. What type of specimen are we using? What are we going to collect from the patients? Where are we going to screen them? How should this be done? We believe that a dental clinic offers a really good opportunity. Once we figure out what that screening test is, a dental clinic could be ideal because men, especially our target population here, we know they go to the dentist regularly. And then biomarkers. There's so much work in biomarkers, and while there are many people working on this right now, there currently is not a concrete biomarker to detect OPC early. And so I put this here because while biomarker work in early detection is really important, understanding oral HPV natural history is key. And if you are not somebody who hears the word natural history often, it was new to me when I started my postdoc. Essentially, this just means understanding the dynamics of an oral HPV infection, from acquisition to persistence to clearance, and what happens, and then that progression to cancer. How long is that? We know a lot about cervical cancer. We know how long women have a persistent HPV infection before it becomes cancer. This isn't evolving. Oral HPV is newer, is the baby in the field. We know a lot about it, but there's still a lot to know in order to have efficient and useful screening and early detection programs. And so I'm going to introduce you to the HIM study, which was a study started by my mentor, Anna Giuliano, in, 2007, I think. So well before I was her postdoc, but it is what I'm going to talk about a lot in this study. It was a multinational prospective study to evaluate HPV in men, the first of its kind, because it specifically was looking at HPV in men. It was in Brazil, Mexico, and Tampa, Florida. They recruited over 4,000 men with no history of these cancers or HIV, AIDS. They began enrollment in 2005, and they followed these men every 6 months up to 8 years with questionnaires and physical exams, collecting genital and anal specimens at each visit, and then testing those specimens for HPV DNA. Well, how does this relate to oral HPV, you might say? About 2 years after this study was funded and they began enrolling, I believe the story was that Anna was on a WHO panel who concluded there was evidence that oral HPV caused OPC. And so they quickly scrambled and added an oral gargle collection to the HIM study and established what we now call the oral subcohort, which is about 3,000 men who have at least 2 oral gargles that were collected and tested for HPV. And so this provides the basis of a lot of the work that I'm doing, as you can imagine. I'm presenting this information at the bottom about general HPV prevalence just to give you a scope. So what they found at baseline, so prevalence of HPV of genital HPV was that over half of the participants had any type of HPV detected. 12% had a high-risk HPV detected. But most interestingly, among virgin men who claimed to have never had sex, high-risk HPV prevalence was 18%. Now, we could debate what's behind that story, but there is one particular question that was introduced to me that comes from this piece of information, and it's what's called a sequential HPV infection. I'm sure this exists with other infections. But this was one of the first things I did when I got to Moffitt, and it was really interesting and a good introduction to oral HPV. And so to define it, so we're all on the same page, a sequential infection is an HPV infection of the same genotype that is found at a different anatomic site after a prior infection at a separate site. And so there are studies that have shown that risk of anal HPV infection was twofold higher among women with a prior cervical HPV infection of the same type. And interestingly, 63% of those women reported no history of anal sex. And so they thought sequential infection. And then the group at Moffitt before my time had looked into prior genital HPV infection and whether it increased the risk of a subsequent anal infection of the same type in men who have sex with women only. And what they found was an increased risk of that subsequent anal infection. And so we thought, why we need to look at this in oral HPV. The same could be true. And so here's a really complicated flowchart that you don't need to read into, but basically what we did is we took that oral subcohort, the men who had at least 2 oral gargles tested for HPV, and we identified men who were eligible to have an incident infection throughout time, meaning they did not have an infection at baseline. So they could acquire a new incident oral infection. And we looked at whether they had a prior genital HPV infection or not. And you can see those numbers here. And so first we just looked at incidence. We compared among those men who had an oral HPV infection, how many of them had no prior genital HPV infection and how many did have a prior genital HPV infection. And really the box that's most important here is you see this incidence rate ratio of nearly 5 when you compare these two groups. And this is specifically for HPV-16. And I draw your attention to this type of HPV specifically. You've probably heard about it in the context of cancer. It is responsible for, I think, about 70% of cervical cancer. But at the oropharynx, it's responsible for 90% or more of all cancer, HPV-associated cancers. So it really is what we're interested in. But we do look at, the high-risk types grouped together as well, and where you see an increased incidence rate ratio. And then, of course, we modeled it and found that when we adjusted for other factors such as age, country, and sexual factors, that having a prior genital infection was associated with an increased odds of the subsequent oral HPV infection. And before I dive into this, I'm going to back up. We did look at that in reverse as well, just to test our theory, and we found the same to be true. So did an oral infection increase risk of a subsequent genital infection? But that's not the key to the story here. And so that was one way that we looked at how men are getting an oral HPV infection. Do we think it tells the whole story? No, but we know that preventing HPV in general is important. Preventing it at the genitals is going to prevent a subsequent infection. So we took another turn, and how can we learn more about an oral HPV infection? As I mentioned, early detection and screening is difficult because there's no precursor lesion. And so what we really need to do is we need to learn more about those infection or men or women with an infection that persists. If we can identify who's at an increased risk for their oral HPV infection to persist, perhaps we can identify a group at highest risk for developing cancer. And so this work was a combination of several different expertise that kind of combined everything they knew. So we had somebody who'd done some EBV work with the nasopharynx, and then we had Anna who had done, HPV work, and then I was doing persistent oral HPV infections. And we said, well, oral EBV, Epstein-Barr virus, really has a lot of similarities to an oral HPV infection. And we know that it's a pretty common infection. Maybe it's part of the story. Perhaps people who have active viral EBV replication are also at risk of having a persistent oral HPV infection. And so just a little bit here about EBV. It's transmitted via the oral contact, so very similar to oral HPV. It's shed in the saliva upon reactivation. It's very common in the general population. Most people have had a prior exposure to EBV, probably never had symptoms, probably never will. However, it does establish a lifelong, persistent, latent infection. So it's always there. You're not going to have symptoms. You're not going to have active disease, but it's always there. The people who are most likely to have concerns are those immuno-incompetent individuals. So someone who has HIV or has immunosuppression, if they have EBV, it could start to replicate and become problematic. And this is true of a lot of herpes infections. But so we tested this theory, and we conducted a pilot study of, I think it was 68 men in the HIM study. So we took all of those people who had a prevalent baseline infection, and we looked at that we had a saved oral gargle, and so we knew what the oral HPV was, and we went back and looked for EBV DNA in that same sample to say, do the people who have higher thresholds of EBV DNA, are they more likely to have an oral HPV persistent infection? We didn't know. Kind of a cool idea to test. And so here you can see where we measured the log EBV at baseline compared to whether they had a persistent infection. So on the bottom, these are men whose oral HPV infection was never measured again. So it was transient, they cleared it. This second group is their oral HPV infection persisted 6 months. And then the final group is their oral HPV infection persisted 12 months or more. And basically we found that just the EBV levels were different between the 12-month persisters and the transient infections, not the 6 months and the transient infections. And so we modeled it, and here we go. Sorry about that. To look at the association of EBV detected at baseline. And we found that while our confidence interval is quite high due to small sample size, that there was an increased odds of having your oral HPV infection persist 12 months or more if there was also EBV detected at baseline. And so, like I said, there was only about 68 men in this study. We expanded our sample size to include about 120, I think, men, and we repeated this, and we're currently wrapping up that study and hopefully to be publishing that soon. So now I'm just going to quickly dive into some oral HPV natural history studies. This is the bulk of what's going on, and I think the cool stuff. So the first thing that we're doing, this paper was just accepted. Myself and my colleague Rachel at Moffitt were kind of leading this work. And so first we just looked at incidence in the HIM study of oral HPV infection. Just a reminder, these men had an oral gargle tested every 6 months for up to 8 years. And so we were able to look through all of those and identify all new oral HPV infections. And so incidence was stable at about 3 per 1,000 person months for each year. And you can see, so over here we compared between the 3 countries, especially since Brazil, Mexico, and US could have very different populations. And this blue line is the US. So we're finding a higher incidence of oral HPV infection in our US participants. But when we compare it by age, there's pretty much no difference, which might seem boring to everybody, but it tells us something important, that men are acquiring oral HPV infection across the lifespan. It's not dipping. We're not seeing no new incident infections in our older population, which is important when you think about targeted vaccination programs. And so then the next thing we did was take all of those people who had an incident infection, and we wanted to look at the duration of that infection. And so this red line here are men who acquired a new infection throughout follow-up. The blue line, I'm not going to go into this, but just for your reference, are our prevalent infections. So these were men who had an oral HPV infection detected at baseline. And what we're seeing is, the duration really was different between these groups, which is not altogether surprising. We don't know how long this group of prevalent infections existed before our baseline study visit. So it could have been an ongoing persistent infection. But we do see the duration, there is still quite a bit of duration among incident infections. When we looked at age, it was marginally significant differences by age groups. I don't have that here, but I just wanted to note that the duration was somewhat different amongst age, which leads us to a story I'm not going to go deeply into here, but is there something happening if men acquire their infection at an older age? Are they more likely to persist? And this is important, especially when you think about their immune response at an older age. We know immune response declines as you get older. That may tell the story, that may not. I will say, interestingly, it's not here on these slides. What we're finding is that when we look at marital status, incidence and duration are higher among divorced men. And so we could dive into that at a later time, but I think you understand what I'm saying. And so we have a lot of future studies. Stay tuned. The next important thing to look at is recurrent and multiple oral HPV infections. It's a complicated piece of the story. Many men had more than one oral HPV detected at a time. Some even had more than one oral HPV persisting at a time. That's a different project, a lot of work. And then what we're working on right now is diving into the lifestyle factors that are associated with a persistent oral HPV infection. So as I mentioned, we have a lot of questionnaire data about smoking, alcohol, detailed sexual history that was taken at every 6 months. And then there's starting to look at things like methylation of known tumor suppressor genes associated with other oral HPV-associated cancers. And then this leads me to my next section of this talk, but using genome-wide methylation to see if we can learn more about those persistent infections. And so with that, I'm going to shift gears a little bit and talk about what I have done thus far to start looking for biomarkers for early detection. So I think I sort of alluded to this earlier, but with OPC, early detection is a good goal because there is no precursor lesion to detect before it becomes cancerous. We do know that most oral HPV will clear in about 1.5 years on its own, but there is a subset that persists and leads to cancer. And that's the group I talked about in the last set of slides that we're trying to learn more about. However, most OPC, regardless of etiology, is diagnosed later when advanced treatment is needed. Again, I said this at the beginning, you're talking about a lot of comorbidities related to the treatment, just due to the location of the cancer. And so this is my case for early detection. Anyways, so while I was at Moffitt, Anna had done some work using epigenetics and DNA methylation, but we really dove into this while I was there because this was one of my interest areas. And so just so everyone's on the same page, epigenetics, I like to share this, is the study of heritable changes in gene expression that do not change the underlying DNA sequence. And so epigenetic changes are those that can sometimes be reversible, and they come with age and lifestyle and environmental factors. And DNA methylation is one such way to measure epigenetic changes throughout life. And so here's my one slide intro into DNA methylation. It does not impact the DNA sequence, but it adds a methyl group, which you can measure and look for changes. We know DNA methylation has a long history with cancer. Alteration of DNA methylation is a hallmark of cancer. DNA methylation is a common cause of tumor suppressor gene repression. And in the context or scope of what I'm talking about here, DNA methylation is of particular interest because there's prior work that has found methylation of HPV-41L3, which is a tumor suppressor gene. The methylation of this gene in the cervix was able to predict cervical precancer and differentiates those patients whose precursor lesion was likely to clear versus persist. And so you can imagine why it might be of interest in the context of oral HPV. And so this first slide is really to just describe some work that I joined into and expanded on while I was at Moffitt. And so something that they had done prior to my time is they looked at methylation of this same gene and HPV-16 in the oral gargle, and they compared that to the tumor and found that it was highly correlated. And so I felt confident they could move forward and investigate this further. And this was about the time that I joined. And so they combined the methylation of HPV-16 and this gene to see if it could predict late versus early OPC. And the goal, I put this here, so to detect early OPC, which is defined as a T1, T2 tumor with little metastases. And so this chart over here, what you can see is that really the methylation of HPV-16 and that tumor suppressor gene did good when you combined all the cases, but really it's predicting late OPC better than it's predicting early OPC, which is fine, but it's not the goal here. And so we wanted to improve that. We said, how can we improve our detection for early OPC specifically? Well, we've done methylation of this one gene. Let's expand it to the whole genome because that's an available tool now. And so we took those same oral gargles in that last study and we tested them for genome-wide methylation. We had a total of 92 early cases, and there were 142 frequency matched controls to the total case population. And our goal was to develop a biomarker panel using the genome-wide methylation. Could we take the single biomarker from the previous slide and add in some additional metrics to create a panel to detect OPC early? And so it was a multi-step process. I'm going to click through because I want the image to show up. And so each of these steps over here on the left corresponds to one of the bars in this picture that you see on the right. And so first we looked only at the Illumina probes, and this bar represents the AUC. It did pretty well. When we added data from that last slide, so the site-specific methylation, our AUC improved. And then we found that just adding HPV-16 status alone, like did they have it, yes or no, improved our AUC even more. And then knowing that we had this site-specific methylation data of the HPV-41L3 gene, we thought, well, surely Illumina, the Illumina genome-wide array, picks up this same gene. And so let's dig into that and pull out those probes individually, which is a long process that you could read about in my paper. But it improved the AUC just a touch more. And so our final AUC on our validation set was 0.935. And so we were feeling pretty excited. We have found this panel that did a really good job at identifying early OPC. So what do we need to do next? And so, sorry about that. So there's a lot of steps, between we've identified it in the lab towards throwing it out in the clinic and screening everyone for cancer. And so this is a framework set out by the EDRN, the Early Detection Research Network, who is involved in a lot of biomarker development. And so these are the steps towards clinical utility, according to the EDRN. It needs to be an easily accessible biological specimen, an oral gargle. Couldn't be easier than that. You need to compare it in cases versus controls. Great, we did that. Now you need to have intra and inter laboratory validity. Well, we couldn't check that box because all of these were tested at Moffitt in the molecular genomics core. And then you also need to have an assessment of confounders separately in cases and controls associated with this biomarker. And so we couldn't do that at the time. Our sample size just wasn't as large as we had hoped. And so we applied for and were awarded a UO1, which is a grant that works with the EDRN to, we're specifically part of a group that is looking at biomarkers for head and neck cancer prevention. And so our specific grant is the biomarker approach to screening for HPV-related OPC. And essentially the aims are to address these 3 unchecked boxes. And so they are recruiting more participants at Moffitt, more early cases, more controls. And then our partner site is the University of Pittsburgh, who is now also recruiting cases and controls so that we can test these samples at Pittsburgh and Moffitt, and then swap them and test them at the opposite place to start to get at this laboratory validity question. And then also just increasing our sample size, we can start to dig into some of these confounders a little bit more. And hopefully have a biomarker panel that works. So with that, I've said everything I need to say today about OPC and oropharyngeal cancer. I'm sure I could share more, but I'm going to end it there and just acknowledge all of the people at Moffitt who were part of this work. As I said, a lot of it was done while I was a postdoc, but a lot of it is ongoing now. I am still doing it and meeting with these people every week to move these projects forward. So with that, I will stop and take questions.
I'll jump in with a question. Great talk, Brittney. It's fascinating. It's really cool stuff. The infectious causes of cancer is always great. I think I asked you this actually when you're interviewing, and I don't remember the answer to it. When you're looking at multi-site infections and you're saying it's the same genotype, how are they determining that?
Yeah, you did ask me this question, and we have subsequently talked about this. So it is, I think basically the answer to your question that would be coming next is no, we don't know it's the exact same virus. So we're using, it was on this earlier slide, it is a test that Anna uses in her lab to detect 25 distinct types of HPV. So we test the oral gargle, they get the viral load out, and then they say this is positive for HPV-16. Right. That's it. That's all we know. So we can't conclude it's the same viral lineage or it's the exact same viral infection.
Does that matter?
You know, it depends who you're talking to. So one of my colleagues is actually really interested in this. There's some fancy word for it, and she makes these trees. You know, is it the phylogenetic trees?
Yeah, I do like
virus. Yeah.
Phylogenomics.
Cool. There you go. And so she's fascinated by that. If you talk to maybe someone like me or Anna, who are primarily concerned in preventing cancer, or just what do we have to do to stop the infection from progressing, all that matters to us is it's an oncogenic type that is still there. We're seeing HPV-16, for example, at visits 1, 2, 3, 4, and 5. That's all that matters to us. And in the story of persistence, no, it doesn't matter. Mm-hmm. We know that we can still call it a persistent infection, even if we don't know that level of detail.
Okay.
Yeah.
That's fascinating. So the other question, if I can monopolize all this time, is these Illumina methylation panels. I think part of my I'm wondering is like how how many target, like when you say like they're looking across the whole genome, is it like known genes that could be associated with something, or it's just like, here's every open reading frame in the human genome and we have methylation?
I think without talking to Illumina, my understanding is it's the latter. They develop a standard set of this like 850K. Well, now it's up to 950 CPG sites, I think, but this work was done on the 850K. It's a standard set of CPG sites. I don't know how they pick them. I don't know how they've dwindled it down to those. I know someone's told me at one time, but no, they're not like, these are the ones that we know are associated with cancer. Now I will say something interesting. So for that grant that I just mentioned to build the biomarker panel, we actually explored, because what you can do is you can do the genome wide, look at all of the CPG sites and figure out which ones seem to be important in the context of your disease. And then you can build, I call it a personal, it's not a personal, a custom panel of just your CPG sites. Well, when we were writing the grant, we reached out, we worked with Moffitt's Genomics Core, and then also with Illumina and one other company, I can't remember. And it was actually incredibly more expensive for us to build the custom panel of our CPG sites than to just run the entire methylation array on every sample.
Like an economy of scale thing.
Yeah. They said they're hoping that it's going to start getting more affordable, because that's the goal with the genome-wide methylation, right? It's not for everybody to do a fishing expedition. That's a great first step, but the goal is to dwindle it down to the important CPG sites. And so we actually realized, like, okay, well, one, it's cheaper, but two, it actually was more standardized, because it's incredibly standardized by Illumina, but then also the people who can run it have to have all this criteria. I'm not doing this justice, but basically they told us it's actually more standardized to do the genome-wide panel. But then the third thing is we thought, well, we're going to be testing a whole bunch more samples, more early cases. What if we identify an additional CPG site that we didn't, at least we have the entire array of data. So.
And then my last question, and then I will be quiet. The hits that you get, right? So they should tell you like the function of the gene that is methylated, right? Does it make biological sense that these would be like precursors to HPV?
Okay, it's funny that you say that they should tell you the function. They don't. They give you a CPG site and a gene, and then me, as the lowly postdoc, spent hours on the Google trying to figure out the function of the gene. And I have a table somewhere, because Anna said the same thing. Well, we need the function of the gene so we can.
It'd be a really nice story if you could like. Put that right
half of them are so arbitrary or like unknown, or I'm like, or it's like incredibly detailed and related with a transcription factor, and we're like, okay, well, I guess that could be associated. Like, so
like a Go analysis or something, like a KEGG analysis, like, oh, here's the genes that matter. What pathways are they a part of? That makes sense.
Yeah. I will say, I think there was at least one where we thought, oh, okay, I can see the story here. Cool. I think at least, let's see, there were 14 markers on our panel. I want to say at least 3 or 4 of them, like when I looked in the gene databases, gave me nothing. It was like, nope, we don't know. And so it.
Cool. Hypothesis generating maybe then, right? Like,
yeah, absolutely. We're hoping that we can fine tune the panel even more as we expand with the current grant. And so maybe it'll get better.
Awesome. Thank you.
Yeah. Thank you for your questions. Okay. I don't think there's any more.
If there aren't any other questions, then we will let everybody go. Thank you so much, Dr. Dickey, for your wonderful presentation today. We appreciate it.
Thank you.